I think we will have a very exciting session because we have some of the best experts in the world on the topic. So I will just show you one slide as an introduction, just to show you how the world of microbiome is expanding. If I can have the slide sometime. Voilà. Okay, so it's just to show you that the number of publications per year is increasing a lot on the microbiota. So you can see that in 1966 we had only 3 publications, and now in 2016, just for the year 2016, there were more than 7,000 publications on microbiome. And you can see that most of the publications are on the topic of the gut microbiota. And you can see that for the skin, respiratory tract, and female genital tract, they are less abundant, of course. And if you do on PubMed microbiome and HIV, you see also that the publications are increasing a lot. And we had 93 in 2016. And here also, the majority of the publications are on the gut and on the respiratory tract and the female genital tract. So this is all what I have to say. And I think now we can start the session. And I have the great pleasure to introduce my co-chair, Alan Landay. Alan is professor and chairman of the Department of Immunology and Microbiology at Rush University Medical Center in Chicago. He has been involved in HIV research for more than 35 years. And his current research focuses on immune pathogenesis and immune-based therapy on HIV disease. And he has also a lot of involvement in the world of the mucosal immune system and microbiome in HIV pathogenesis. So, Alan.
Thank you very much, Elizabeth, and I'd like to thank everybody for coming to this session. I think it'll be very exciting, and I've been tasked with giving the overview today of the sort of more of a global overview, because the organizers had asked, many of you may be new to the human microbiome and studies. I know at an IAS meeting, this is one of the first sessions at an IAS meeting that I'm aware of devoted entirely to the microbiome. I think it's been a topic in many of the sessions previously as individual lectures, but now we're really focused here totally on the microbiome. I show this first slide because I think it gives you an idea that the microbiome is really out there in the public domain, and you can look at on the left-hand side here in The Economist about microbiome, and in the Sunday New York Times about the role of germs. And again, all of you should be having a dog if you have young children. They're good for the microbiome. Cats aren't, I've been told, but in terms of allergic disease. So you may be a cat or a dog person. But I think this just points out to you in both the scientific and the lay literature, there's a tremendous interest out there. So I think the fact that now in an HIV session we're beginning to focus on the human microbiome is very important as we understand its role. I have no conflicts on this talk. If we look at the human microbiota, we really call it a superorganism. And I think one of the important points is we're dealing in the microbiome with 100 trillion cells versus 10 trillion cells in humans. So we're more, one of your cocktail hour facts that you can take back when you have dinner tonight with your friends or colleagues is that you're more microbial cells than you are human cells. And I think that's an important fact we all need to recognize, and the importance of the human microbiome across all the sites in the body that we look at. There's also the number of genes are much greater, in 3 million genes in the microbiome and 23,000 genes in humans. So the important role here is it has important in health, and as we go along also to disease. And we'll be talking and focusing today specifically on the role of the microbiome in HIV. I think one of the critical factors that we face, and you'll hear a lot about this, is what are the methods to study the microbiome? And one of the adages I've heard now coming at a number of meetings, it's just not who's there, but what they're doing. And I think that's another important thing to take away from a talk like this. It's looking more at the functional outcomes. So you'll hear about which microbes are there, and we're using predominantly the 16S RNA to do that. But now we're moving on more to the metagenomics, metaproteomics, and metabolomics. So we're beginning to look more at the functional outcome and the metabolome of the microbiome, and I think that's a critical part of where the future research efforts are going in the field of microbiome in general and in HIV specifically as well. There has been, as you're aware, a human microbiome project. There's also, that's in the U.S., funded through the National Institutes of Health, and there's also been European microbiome projects. But I show this slide really to point out the variety of sites we can study the human microbiome across, from the skin to the nose to the oral cavity to the GI tract to the female reproductive tract. So the microbiome will vary across all of these sites as we look across here. And on the bottom, looking at the metabolic pathways, it just shows you across each of those sites the variability one sees. So each site is different in terms of the predominant of the microbial types. No core microbiome at every site for every one. And I think that's the whole question that we raise is, as we look at the human microbiome across the human populations of the world, it'll vary, and that's a critical part. As an HIV meeting, we do study HIV as a global disease and need to understand the concept of how the microbiome varies across our global populations. And importantly, it's also about the functionality of the microbiome. As people think about the microbiome, most of the times we think it's bacteria, and that's what we're going to sequence. But it really now we're looking well beyond bacteria. Earlier in the meeting, Doug Kwan gave a talk on the work on the virome work that he's been doing, and others have been starting to focus on the human virome and the role of the virome, both the prokaryotic and the eukaryotic virome. So I think there's an important role now that as we think about studies in HIV and the microbiome, we don't limit ourselves to solely bacteria, but we begin to expand this, because there's an important role. As we know, bacteriophages are those that are found in our bacteria, and as we look at those viruses, the prokaryotic viruses, we need to understand how do they affect function, how do they affect outcome, and how do we study that. And that, again, is going to evolve a whole different level of analytic capabilities and abilities to look at this. Beyond the virome, we have the microbiome, and the microbiome involves the fungal organisms. Again, across all of our different anatomic sites, whether it be in the oral cavity, in the lungs, in the gastrointestinal tract, or in skin, we can find now a diversity and looking at this, that it's disrupted and changed, and that there is a real important balance between the fungal communities and the bacterial communities in the host. We're just beginning to study this, and I think, again, just bringing this up as an important area for evaluation is where we want to go further. So what do we now know about the gut microbiome in health? Well, first of all, we have over 10 to the 14th organisms in the gut intestinal tract, making us almost 90% bugs and 10% us. As I said before, we're a lot more microbiome than we are human. The microbiota is the collective microorganisms, and it's also the gene content. I think that's the critical part. It's not just classifying the actual species and genus and species of the organisms, but also what are their genes and their function. And now we also use a term which many of you have heard about, dysbiosis, a dysregulated or abnormal microbiota, common in many diseases. And so really the first of those studies that Elizabeth pointed out in the literature relating to diabetes, colon cancer, inflammatory bowel disease, a lot of that original work was done there. We're now moving into studying more about the role of HIV, and it's really, we've come new, as you can see by the numbers of publications, to the microbiome field from the HIV field. And we are looking at site-specific microbiota in the gastrointestinal tract. So those people who study, as we'll talk about many sites today in our symposium, we need to understand they are going to be different, and we need to understand that relationship across multiple sites. There's also an important aspect that across the GI tract, the composition varies in terms of both the numbers and who are the microbiome composites. When we look at the actual colonic content of the microbiome, we're looking at 10 to the 11th to 10 to the 12th, whereas in the small intestine, in the duodenum, 10 to the 2 organisms. So quite 10 log differences in terms of the numbers of organisms across the GI tract. This brings up an important question of how do we sample and what do we sample in the human GI tract? Do we look simply at fecal material, or do we begin to now start to do gut biopsies across these various sites? I think that's critical because there is variability, and we need to begin to understand that further There have been some studies in this area, but I think many more are needed. And I think there's some great opportunity for folks here in the audience today in terms of this and really beginning to get into this area of research. There's also an important role for the gut microbiota across the human lifespan. And as we're studying HIV, we know this is a disease we study from birth all the way through to adulthood. And now we're beginning in normal microbiome. There is clearly a difference in the diversity across the human lifespan, as you can see in the top part. And as we get into aging, in the aging populations, as we're now seeing older HIV populations over age 50 and age 60 in our clinics and populations across the world, we're going to be dealing with inflammation, inflammaging, and other aspects of immune senescence in a healthy aging population as our HIV patients, and also the potential impact of HIV in addition to that on the human microbiome. So there is clearly changes, as you can see, in terms of diversity. When you're born here, you have very low diversity, which you then meet higher diversity in midlife. And then as you age, over normal healthy aging, you also reduce the diversity. So we have to overlay now in this normal course of changes in the human lifespan of the gut what happens in HIV disease and begin to integrate those components as well. And I also want to raise a point here about the differences in populations across the world. The Western microbiota diverges from that of non-Western, and this is a paper that was published a few years ago and just showed very clearly that a U.S. population was very different than an Amerindian population in South America and a Malawian population in Africa. So as we begin to think about the global epidemic of HIV, there's also a global aspect to the microbiome. And we're just beginning, as I think was shown again by Doug Kwan in his lecture, by showing a map of the world, that there are really significant differences across the globe in terms of the human microbiome and diversities as shown here in this slide. So when we think about then what happens in HIV and what's happening here, what I'll focus on is the gut in HIV. So overall, in an overview, there's differences in the gut bacteria diversity, taxa composition, and metabolism have been reported. There's also an important role, perhaps the normal microbiome is not restored in all subjects on ART, and again, variable data there. We've also begun to understand that the ARVs themselves may have an impact and induce microbiome changes. So I think we begin and have to think not more in cross-sectional but longitudinal studies, looking at patients pre- and post-therapy to understand the impact of the microbiome. We're also looking at prominent shifts in Prevotella and Bacteroides, and I'll get more to the functional consequences of that. And those have been associated with the men who have sex with men designation. So there may be in high-risk groups changes in the microbiome independent then of HIV infection. We begin to need to understand that and the role of the sexual behavior in our populations and how that might affect the outcomes of our microbiome studies. We also have shown, and it's been published, that dysbiotic bacteria are shown to translate in progressive SIV infection, and also there's dietary changes that are there that can also be associated with disruption and HIV infection. So that's another important part, is what are the diets that our populations have that really influences heavily the microbiome changes, whether you're on a Western diet and you go to McDonald's every day or you're eating a vegetarian diet. So those can all effectively impact the host microbiome. One of the things that we're interested in is the role of the immunoregulatory microbes and in the immune system interaction. What we have seen in our studies, and many folks have shown this, that there's increases in bacteria with are called pathobionts that are predominantly Prevotella or Acinetobacter. And then there are decreases of bacteria that make anti-inflammatory mediators, especially those of the short-chain fatty acids. So we're beginning to focus a bit of effort in this area of understanding the role of short-chain fatty acid. The prototype for this is butyrate. It's a fermentation product of insoluble dietary fiber, an energy source for colonic epithelial cells, and therefore an important role in maintaining gut barrier integrity. So it's a marker and a molecule that's critical for helping to keep the gut from becoming leaky, as we've seen in the HIV populations. And also, it's important in regulation of Treg differentiation, APC, and T cell function. So there's an important role in the immune system. And we're beginning to look at the interactions then of these products from the bacteria in the gut and other sites and how they interact now with the immune system, but not just with the immune cells in the gut, but also epithelial cells, which is another critical part of our evaluations. We can't lose sight of the fact, especially in the gut and other sites in the body, the epithelium is a critical part of the immune and functional outcome. We're also interested in thinking more about the therapeutic role of the microbiome in HIV, and you'll hear more about that from Rupert Call today. This is where we want to restore the equilibrium. We're thinking about prebiotics, which is basically the food for the bacteria, probiotics, and we've done a number of studies, and folks here, and you'll hear from Dr. Klatt and Dr. Brenchley today, who also had worked in the primate model in doing therapeutics in the role of probiotics in the monkey models, and we'll be talking about other things today, but there have been a number of studies started in that area. We're also thinking about postbiotics, the role of butyrate and other short-chain fatty acid metabolites, how they might affect functional outcome. Or we can combine pre, pro, or postbiotics, which is known as synbiotics, so combining these together. And then finally, one of the newer areas that's had a lot of interest in the Clostridium difficile infection area, looking at fecal transplants, and those have already begun both in animal model and human trials in HIV. So I think there's a great opportunity to take what we've learned, and we'll hear a lot of that today from our speakers, to try to integrate all the features of moving forward in a therapeutic approach. If we're now looking at this, this is a model that was put together by Steph Dillon and Carol Wilson. I've borrowed from their review in AIDS, but just to show that there's an increase in these bacteria that are the pathobionts, a decrease in immune regulatory bacteria. That can lead to this increase in microbial translocation that was characterized a number of years ago, that can then activate innate lymphoid cells, important immune cells or innate cells in the gut, and then causing inflammation and eventual T-cell activation and depletion. So there's a lot going on at the GI tract that the microbiome may initiate, and HIV may be an important role in, and we're just beginning to understand these basic mechanisms, and we'll hear much more about this today. So finally, I just want to talk about future directions. I think as an audience today, thinking about the role of the microbiome, there's a lot more work we can do. I think studies on metagenomics, metatranscriptomics, and metabolomics are key, and I hope in the next years that at these meetings we'll be presenting results of such studies. We also need to extend our work to the virome and microbiome, define the interactions of the gut microbiome with the mucosal immune system and epithelial cell biology. I think that's critical because it's not the microbiome alone. It's an important interactive role. And then finally, we need to move beyond the gut, and we'll hear a lot more today in the following talks about the female genital tract microbiome. We also need to think about the lung, oral, skin, breast milk microbiome. All of these are separate and important and play a real functional role in our better understanding for HIV infection. And I'll just end there and thank some collaborators who provided information for the talk from the University of Colorado and University of Alcala. Thank you again for your attention.
Thank you very much, Alan, for this very beautiful review. We have time for one comment or one question from the audience. Okay, so we will move to our second speaker, Jacques Ravel. So Jacques is a professor in the Department of Microbiology and Immunology and the Associate Director for Genomics at the Institute for Genome Science at the University of Maryland School of Medicine in Baltimore. We were very lucky to have him in France for several months, even 2 years now. Two years. And so he will speak today about the microbiome in the vagina and HIV infection. Thank you, Jacques.
Thank you very much, Elizabeth and Alan, for inviting me to speak in this session. If I can have the first slide. Do I do it here? Okay, good. So I was kind of tasked here to give you a little introduction on the vaginal microbiota and how it can play a role in HIV disease. So just to start, I have absolutely no conflict for this talk. And just to give you a little introduction on the vaginal microbiota, lactobacillus, which are species of bacteria that produce lactic acid, often, but not always, dominate the vaginal microbiota in reproductive age women. And that's a very important characteristic of the human microbiota. And this is not something we'll find in any animal model that we know of so far. And we've looked, in my lab and others have looked at a lot of animal. They do not have this characteristic. They don't carry lactobacillus, and they have a lot of different anaerobes, and they have a pH that's often close to basic. So because lactobacillus produce lactic acid, they acidify the vaginal microbiota to a pH which is traditionally around 4 and less than 4. So a very acidic environment means certainly that it's a very unhospitable environment for non-indigenous microbes that try to colonize the vaginal tract. So this is a mechanism that is believed to be important in which the vaginal microbiota play a role in protecting against disease. An interesting aspect of the vaginal microbiota is that throughout a woman's lifespan, it tends to change quite dramatically from birth to pre-puberty. At puberty, we enter the reproductive years where we see lactobacillus, and then at menopause, we have another change. And outside the reproductive years, lactobacillus is not present in the vaginal microbiota. So a very dynamic environment throughout a woman's lifespan. So we characterize and describe the composition of the vaginal microbiota a few years ago. And this was done through a pretty well-powered study where we had 400 women. Does this work? We had a pretty well-powered study where we had 400 women. And what was interesting about that study is that the participant represented 4 different ethnic groups equally. We had Asian, Hispanic, Black, and Caucasian women. And what you can see here is that each vertical line here represents one woman. And the composition of a woman microbiota is indicated by the color of each one of those 25 most abundant taxa here. And you can see that yellow means it's pretty much absent, and red means it's close to 100%. So when you cluster those different microbiota, what you see is that you can cluster those microbiota in 5 what we call most prevalent microbiota type. And we call them community state type. Okay? And we have 4 of them that tend to be dominated basically by one of the 4 species, wrong side. One of the 4 species of lactobacillus, which is Lactobacillus crispatus, Lactobacillus iners, Lactobacillus gasseri, and Lactobacillus jensenii. And then we have a 5th type here, where we basically have an absence of lactobacillus, but instead a wide range of anaerobe, facultative, and strict anaerobes. Okay? And this anaerobes represent about 25% of all women in this particular study, which is quite impressive to me. And a lot of people would consider this state diseased. And I don't think we can say that 25% of the population is diseased. So I like to think about that state a little differently, and I'll get to this in a minute. But what was interesting about the study is that, like I said, is that we had the 4 different ethnic groups, and we were able to look at the frequency of those different community state type across the different ethnic groups. And one remarkable finding is that if you look carefully at Black and Hispanic women, you can see that about 40% of those women actually carry a microbiota that does not have lactobacillus. So again, a very important fact. And we cannot say that 40% of all those women are actually diseased. In white women, we have Lactobacillus crispatus being the most prevalent microbiota type. And in Asian women, we tend to have Lactobacillus iners in yellow, in orange here. That's the most abundant type. So, like I mentioned, I'd like to think about this microbiota. When we look at this, we see one time point in time. Okay, it's one characteristic. And we didn't know anything about how this microbiota was going to change over time. So it's possible that in one day you are one type and another you're another. So to test this, we actually performed a study where we enrolled 160 women who sampled themselves for 10 weeks on a daily basis. So a very intensive study to perform. And when we do this, we basically were able to observe 3 major types of longitudinal profile of change of the microbiota. The first type was something like this, where a woman tends to be dominated by one lactobacillus. Here is Lactobacillus crispatus in red, and it will be throughout the talk. And in orange is Lactobacillus eners. So you can see that the microbiota is dominated close to 100% at all time, except during menses. The first woman is dominated by, or there's a spike of Lactobacillus eners during the 2 menses event, and in this woman we have a spike of Streptococcus. So we can see the same type with Jensenii and Gasseri as well. Then a second most interesting set of profile was those guys, where we see somewhat of a stability over time, but we do not see Lactobacillus, which was the most surprising to us. I would have expected that the state where you don't have Lactobacillus would last for a few days or maybe a week, but then we'll come back to Lactobacillus. But no, those women maintain basically an absence of Lactobacillus, and they were basically dominated by organisms like Garnerella vaginalis or Atopobium vaginae, and I'm sure Nikki will talk about those and their importance later on. And then the third type was something more erratic. Those women basically did not have a consistent type over time. There's a constant change basically on a daily basis from one type to another. You can still see some Lactobacillus here, crispatus and eners. They look like they're ecologically fighting over trying to dominate this microbiota, but can never do it. So 3 major types. But we were very interested in those kind of microbiota and what's their importance in sexually transmitted disease, including HIV. And this is how I think about those. We call it community state type 4. And I like to think of it as a normal state that carry risk. So we know that at any given time, in America, at least, it's 25% of women don't have lactobacillus. If you go to Africa, it could be up to 50%, depending on the region. So this state is very interesting because it's associated with what we call a very high Nugent score and higher pH as well. So Nugent score is a gram stain where we evaluate the presence of morphotype that looks like lactobacillus, or those that look like Gonorrhea, or another type called Mobiloncus. And it's used in research setting for the diagnosis of bacterial vaginosis. It's a very interesting test. It's very easy to do, very easy to use. It can be done prospectively, and we can do this in a very large number of samples. So those studies have been performed, and what we've shown is that you have very high Nugent score tend to be associated with increased risk of HIV and other sexual transmitted infections. So we have women that appears to be asymptomatic at most time, and they are apparently normal. Maybe we should not be used the word not normal or abnormal. Maybe not optimal might be a better terms when it comes to this sexually transmitted infection, but potentially an increase of sexually transmitted infection. So we looked into the role of cervical vaginal mucus as well as the vaginal microbiota in the context of HIV. So we know that HIV must penetrate that cervical vaginal mucus, which is that first line of defense to reach the target cells. And we hypothesized that cervical vaginal mucus has the ability to retard or immobilize the HIV variant. And if it can do this, it can actually reduce or even prevent the initial infection. So we wanted to test this. And to do this, we performed an experiment where we collected 31 cervical vaginal mucus from women. And we measured the mobility of HIV variant, that are fluorescently labeled, inside this cVM, so under microscopy. And so you can see here, we obtained two different groups. One that had 14 women, where you can see the variant moving. And so this group was called the mobile variant. And then we had the second group where basically the variants were trapped into that cervical vaginal mucus. So we tried to explain the difference between those two groups. And we looked at different things. And we looked at pH. Of course, pH, if you have a higher pH, you have a higher Nugent score, you should be more susceptible. So when you take all the sample, this is the case. If you reduce yourself to sample that have what's called an healthy pH of less than 4.2, you lose that difference. With Nugent score, it's the same thing. High Nugent score associated with mobile variant, while low Nugent score with trap variant. And then if you go to a Nugent score of less than 3, there's no more difference. When you look at total lactic acid, it's the same thing. Obviously, more lactic acid, lower pH. As soon as you go to a higher concentration of lactic acid, you lose the significance. So we can conclude that pH, Nugent score, total lactic acid couldn't explain those differences. So then we looked at D-lactic acid or L-lactic acid, because lactic acid is produced in 2 different isomers. And what we found is that actually with D-lactic acid, there was a higher abundance of D-lactate was associated with trap variant, and that difference stayed if you go to higher concentration of D-lactic acid, while it didn't show this pattern for L-lactic acid or for the ratio of D and L. Why lactic acid is important? It's because lactobacillus produce lactic acid in differential amount. And depending on the species, and I should say even on the strain within the species, you can see different amount of lactic acid. And you can see crispatus tend to produce a lot of D-lactate, just like Lactobacillus jenseni, while Lactobacillus iners doesn't produce any lactic acid. And even if you do metatranscriptomics, which is what Alan was talking about, where we looked at the genes are activated in the microbiota, we can see that D-lactate dehydrogenase, which is necessary to transfer to make from pyruvate to D-lactate, is not expressed in Lactobacillus iners, while it's expressed in Lactobacillus crispatus-dominated community. So we characterized the microbiota of those samples, and what we found, we were able to group them. They're not communicate type, but group them in 3 groups, those that are dominated by Lactobacillus crispatus, Lactobacillus iners, and those that had a mixture of other type. And this was able to explain very nicely the pattern that we saw for the mobility of the variant. With group 1, the crispatus, where you see absolutely no mobility, and mobility increases as you go through iners, intermediate, and no basically very little lactobacillus, the highest mobility. And this obviously stayed for pH, Nugent score, total lactic acid, and D-lactate, again, was a factor because Lactobacillus iners does not produce D-lactate. So I just want to finish with just a few perspectives. I think we need to redefine and come up with a better definition of dysbiosis of the vaginal microbiota. I think this is going to be necessary if we want to move forward as a field, because bacterial vaginosis, which is what's being used right now, is a really ill-defined disease, and a condition, I should say. And we don't really have a good grasp microbiologically or metabolically what this disease represent. So, and also because bacterial vaginosis is a state, it's one of the states of the vaginal microbiota that carry risk, that we know this because it's associated with high Nugent score, but it's not the only one. And just as I showed you with Lactobacillus iners could be actually a risk factor that might be underestimated. And treating BV appear to be inefficient in eliminating the risk of transmission of HIV or other STI. So we need to expand our understanding not only of the microbiota itself, but the mechanism by which it provides that first line of defense, and use that information to develop better strategies. And not only develop them, but implement them. We need to think of ways, maybe chemically, but also microbiology, try to manipulate to restore the vaginal microbiota. I think this is an aim that needs to be done, and it has to be a solution that is widely distributed and can be taken by everybody very quickly. And with that, I just want to thank some of the collaborators, especially Sam Lay, with whom I collaborated on the project on the cervical vaginal mucus. And with that, I'll be happy to answer any questions.
Thank you very much, Jacques, for this very fascinating presentation. I am sure there are some questions in the audience. Yes.
It's a general question. Laurent Kaiser from Geneva. Is there a link between the microbiota you find in the vagina or in the gut in these people, or it's separate compartments?
Oh, you mean, is there a link between within a person, her
Yes.
vaginal and gut microbiota? It's pretty unclear. There doesn't seem to be one. But if you think about bacterial vaginosis, so you can think that maybe having a dysbiotic gut microbiota could lead to a dysbiotic vaginal microbiota. That's entirely possible. I truly believe there's a gut-vagina axis, just like you have a gut-brain axis.
Thank you for the talk. Have you looked at whether or not there's a pattern or an association of the women who are on birth control, considering the fact that certain women who are on birth control seem to have more chronic levels of inflammation, and whether or not that also affects the microbiota?
Yeah, we have a study ongoing right now where we looked at women initiating birth control. So inflammation that's a generalization that's only applied for certain type of birth control, like depot or things like this. But other pills, and like especially estrogenic pills, it's the opposite. It's, you see an association with higher level of community state type dominated by lactobacillus as opposed to non-lactobacillus dominated community. So women who initiate them, if they have a microbiota that doesn't have lactobacillus, as they go on estrogenic pills, then they tend to regain lactobacillus dominance.
Hi, Kelly McDonald. I'm just wondering, looking at these women as you have, looking at ethnicity, but not looking at their risk factors for STIs, for rates of partner change, for other inflammatory factors. You and I know there's a huge list of demographic variables that would influence all these factors just as much as ethnicity. Have you done that so that you can tease out what has an actual impact on this? Because that obviously would really confound the data.
Absolutely, and I want to rectify. I'm not claiming that ethnicity or genetics is driving solely the patterns that we saw. Absolutely not. That's not my point here. And you're entirely right. There's certainly, for example, douching is much more prevalent in Black American women than it is in white American women. And there are studies that shows it's associated with loss of lactobacillus. So there's a lot of other confounders, and the study was not well powered to answer those questions. So we tried to control for as much as we can in the statistical analysis, but we couldn't control for everything without losing power.
Okay, the last question over there.
Jacques, that was a terrific talk. When we talk about gut dysbiosis, we often measure it against kind of the norms of what we find in the U.S. or in U.S. white women as compared to either women of other races or women of other geographic locations. And my question is really what we call dysbiosis. What could be the possible functionality of that genital tract that may exist in a geographic place that's quite different than what we may see here? So we call it dysbiotic, but it actually may be functional in some way. Can you say a little bit about that? I mean, there's a reason why it looks this way.
Well, I think dysbiotic is kind of it's a weird term that people have developed dysbiosis, and we don't really know what it means, really. But I guess what you're getting to is the fact that even in what we call dysbiotic, there might still be some functions that are maintained that still could lead to protection. Yeah. And that's what we would need to use. So definitely, what I mean by redefining, dysbiosis is really coming up with a different definition that's maybe based on functions or metabolites or other things.
Thank you again for an excellent talk. Our next speaker will be Dr. Jason Brenchley. Jason is a senior investigator at the NIAID-NIH in Washington, D.C. Jason has a long career in working in the microbiome and HIV area and has done some very important work early in his career on defining the role of microbial translocation, and has moved that work now into some of the important novel studies of studying the microbiome in animal models, which he'll be presenting to us today. Jason?
Thank you very much for the introduction, and I'd like to thank the organizers, of course, for the opportunity to present some of our recent data related to the composition of the microbiome and whether or not and how if it plays a role in the disease progression. So I hope it goes without saying that HIV-infected individuals, irrespective of whether they're treated with ARVs, have a hyperactivated immune system. And part of that hyperactive immune system is attributed to a phenomenon that we coined several years ago, over a decade ago really, that we called microbial translocation. And that referred to the translocation of bacteria. This is a colon section from a chronically SIV-infected Asian macaque, a translocation of luminal bacteria from the lumen of the GI tract into the lamina propria, and that was attributed to focal areas of damage to the structural barrier of the GI tract. And then once the microbial products got access to the lamina propria, they can really go systemic and activate the immune system. So there are certainly abnormalities within the HIV-infected and SIV-infected host that are allowing microbial translocation to occur. There's massive depletion of CD4-positive T cells from the gastrointestinal tract. There's preferential loss of IL-17 producing CD4 T cells and other lymphocytes from the gastrointestinal tract. You can see that there's damage to the structural barrier of the GI tract, so those are all facilitating microbial translocation. But over the course of the past several years, there have been several groups that have suggested that the composition of the microbiome in an HIV-infected individual might actually be at least partially facilitating microbial translocation. So there is actually pretty good consistency with what types of microbiomes exist in HIV-uninfected and HIV-infected individuals. So this is from Stephanie's paper from a few years ago, and she was looking at the composition of the microbiome in HIV-uninfected individuals, so this is fecal microbiome, and contrasting that to HIV-infected individuals. So one of the big differences that she saw, these red and green, so these are Bacteroides and Prevotella. You can see in HIV-positive individuals there is an expansion of Prevotella. Now these blue and orange are the firmicutes, so these are gram-positive organisms that make things like short-chain fatty acids that Alan was talking about earlier. And you can see that the firmicutes are decreased in frequency in the GI tract microbiome of HIV-infected individuals. And you start to see expansion of these gram-negative bacteria, so these are proteobacteria. And a lot of these aggressive bugs are things that we kind of think of as being pathogens like Pseudomonas and Yersinia and E. coli. And you can see fairly routinely, if you look at the composition of the microbiome in HIV-infected individuals, you see alterations to the Prevotella Bacteroides ratio, collapse of the firmicutes, and expansion of the proteobacteria. So we wondered whether or not we would see the same thing in a non-human primate model of progressive HIV infection. So remember, when we're studying cross-sectional studies in HIV-infected individuals, you're looking at HIV-infected people and HIV-uninfected people. One of the key advantages of the non-human primate model is that we can actually follow the same animal longitudinally. They are in the same cage, getting, for all intents and purposes, the same diet day in and day out. So what we're looking here is about 7 or 8 SIV-infected Asian macaques before they were infected on the left, and then during the acute phase of infection on the right, day 21, which is really when inflammation is going rampant. And here are the Bacteroidetes, here are the firmicutes, and this tiny blue fraction are the proteobacteria. So when we average the composition of the microbiomes of the animals before they were infected and day 21 post-infection, you can just see that there is absolutely no hint that SIV infection itself is facilitating a change in the composition of the microbiome. Now that begs the question, why is it that HIV-infected people are looking dysbiotic compared to HIV-uninfected individuals? And in a very recent study, a group started trying to control for specific demographics, and Alan referenced this study earlier. And what they had concluded is after, I don't know if you can read this here, but after stratifying for risk factors for HIV, most of the dysbiosis that is observed in HIV-infected individuals disappeared. So that doesn't tell us that dysbiosis is important or unimportant for the disease, but rather what is causing the dysbiosis that is observed in HIV infection probably isn't the virus itself. But when we're looking at the composition of the fecal microbiome, that just tells us who's there. That doesn't tell us anything about what they're doing. So in a recent study, we actually extracted bacterial DNA from 2 distal sites of either chronically SIV-infected rhesus macaques or chronically SIV-infected pigtail macaques. And again, we looked in liver and mesenteric lymph node, and then asked the question, which bacterial taxa were more prone to be within the lumen of the GI tract compared to these anatomical sites, and which bacteria were more prone to be in the tissue as opposed to the lumen of the gastrointestinal tract. And what we found is that the composition of the fecal microbiome didn't really predict at all which bacteria were translocating. And in fact, if you look at the bugs that were not translocating, there's the Prevotella. So one of the major components of the microbiome was staying within the lumen of the GI tract. If you look at the bugs that are translocating, you can see a couple firmicutes, but these are staph and strep. We think these are contaminants for when we actually, from the skin, from when we opened up the animals. But the bugs that are translocating are really the proteobacteria. And it's down to even one family, Burkholderia. Burkholderia is a tiny fraction of the fecal microbiome, and yet these are the bugs that tend to be translocating to both the liver and mesenteric lymph nodes of 2 species of SIV-infected macaques. So the question was then, why was it that the Burkholderia were preferentially translocating? Were they just coming from maybe the small intestine and they were more prone to be in the small intestine? So we actually did a survey and looked at the composition of the microbiome in non-human primates that were SIV uninfected, starting at the duodenum, moving into the jejunum, into the ileum, and as soon as we get to the cecum, you can see a huge difference. So the composition of the large intestinal microbiome, over here is feces, doesn't reflect at all what's happening within the small intestine. This big orange bar is actually proteobacteria, but it's not Burkholderiales, it's all Pasteurella, and it's not there at all when you get to the large intestine. The Burkholderiales that we found was only in the colon. It wasn't present at all in the small intestine. And also, I just want to point out that the non-human primates don't have the genus Bacteroides so far as we can tell. So if there was an important Bacteroides Prevotella shift, we wouldn't be able to see it anyway. But that, again, seems to be attributed to risk factors for HIV. So the question now was whether or not induction of dysbiosis might affect disease progression in SIV-infected Asian macaques. So our hypothesis was, in the context of increased microbial translocation that is certainly caused by HIV and SIV infection, if we induce experimentally GI tract microbial dysbiosis, would we make it worse? Would we increase microbial translocation? Would we increase systemic inflammation? Would we enhance the loss of GI tract resident CD4 T cells and Th17 cells and possibly accelerate disease? So the way that we changed the composition of the microbiome was with a specific antibiotic, vancomycin. So this is the composition of the microbiome in uninfected animals before they were treated with vanc. Here's the firmicutes, a lot of Bacteroides, a tiny amount of proteobacteria. When you give them vanc, which is a potent gram-positive inhibitor, you can see that just 1 day post-vanc, you go from 4% proteobacteria to 19% proteobacteria. The firmicutes collapse, the Bacteroides collapse a little bit. And then at day 7 post-vanc, it gets even worse. So 25% of their fecal microbiome becomes proteobacteria with a huge collapse in the firmicutes. So the question was, if we give monkeys this microbiome and try to keep them with this microbiome and then infect them with SIV, what happens? So this is the study layout. We looked at everything that we know how to look at. We looked at the composition of the stool microbiome. We looked at different aspects of the immune system and peripheral blood mononuclear cells. We did rectal biopsies, jejunal biopsies, lymph node biopsies. Before the animals were infected, before they were treated with vanc, we then started giving them a dysbiotic microbiome before we infected them. We gave them 2 months worth of vancomycin. We infected them with SIV, and then we, again, studied as aggressively as we could all of these different parameters. We had animals that were treated with vanc. We had animals that were control. The first thing that we looked at, of course, were the plasma viral load and the number of CD4 positive T cells in peripheral blood. The animals in green are always going to be the animals who had vancomycin. The animals in closed circles are going to be always the animals that were controlled. You can see that both groups of animals lost CD4 positive T cells from peripheral blood, but the vancomycin-treated animals certainly didn't accelerate loss of CD4 T cells from peripheral blood. We looked at plasma viremia. There's the acute viral load into the set point, into the chronic phase of infection. There was one animal that progressed fairly rapidly who ended up getting VANC, and his viral load was about 10 to the 9th, so that's what this blip is attributed to. But you can see overall throughout the chronic phase of infection, the viral loads were virtually overlappable between the animals that were controls and the animals who got VANC. We looked at loss of CD4 positive T cells from the small intestine by jejunal biopsies and the large intestine by rectal biopsies. And again, you can see massive and preferential loss of CD4 positive T cells from both anatomical sites. But again, the VANC-treated animals are overlapping completely with the control animals. We looked at the frequency of Th17 cells, which are important for maintenance of the epithelial barrier in the small intestine and large intestine. And again, you can see that both animals lose Th17 cells from both of these gastrointestinal sites, but the vancomycin-treated animals are just not losing more Th17 cells than the animals who had a normal, healthy microbiome. And then lastly, we actually looked at how much microbial translocation they had. We looked at systemic markers of microbial translocation. One is called intestinal fatty acid binding protein, which is made by epithelial cells in the GI tract. When there's epithelial loss, the levels of IFAB go up systemically. We also looked at soluble CD14, which is a marker that is made by monocytes after they get stimulated by LPS before the animals were infected into the chronic phase. And you can see that there were slight differences between the VANC-treated animals and the control animals, but it did not approach statistical significance. And then finally, at necropsy, we can actually do an immunohistochemical analysis for microbial translocation. So this is a colon section, this is the lumen, there's a lot of bacteria here, and you can actually quantify how many bacterial products are within the lamina propria of the colon. You can do the same thing in the mesenteric lymph node and liver. So we did a pretty extensive analysis of how much microbial translocation control animals or animals who had VANC had at necropsy, and you can see in either the colon, the mesenteric node, or the liver, the VANC-treated animals just did not have enhanced microbial translocation compared to animals who were SIV infected, who also had microbial translocation, but the VANC didn't make it worse. And then lastly, we actually followed the animals all the way until they progressed to simian AIDS. And you can see in this Kaplan-Meier curve, the VANC-treated animals just did not progress to AIDS any more rapidly than the animals who had a control diet. And they also died of the same types of opportunistic infections. So there was just nothing that the dysbiotic microbiome was doing to the progression of the SIV-infected animals that we could tell. So in conclusion, GI tract resident microbes, we absolutely believe contribute to SIV and HIV disease progression. They absolutely translocate from the lumen into peripheral tissues and exacerbate inflammation. Even in ARV-treated individuals, and this is important, residual inflammation occurs, and we think that's associated with microbial translocation, which has also been shown to be associated with increased mortality. In the absence of overt dysbiosis that we see in SIV infection, it's really only specific microbes that are preferentially translocating of that order Burkholderia. And dysbiosis of the GI tract microbiome certainly occurs in HIV-positive individuals, but the extent to which it's attributed to the infection, I would say, is a little bit unclear. Probably most of it is risk factors for HIV. And induction, experimental induction of GI tract microbiome dysbiosis just does not influence untreated disease progression in Asian macaques. So I'll close there and just say that we don't know whether or not microbial dysbiosis might have a role for residual inflammation in ARV-treated individuals, and it's also quite possible that dysbiosis might influence susceptibility to infection, and we've got experiments ongoing to try and address that. And I'll just finally close and thank the individuals who did all of this work. Most of this work was done by a phenomenal staff scientist in my lab, Alex Ortiz, who has this data and much more at poster 132. And when we have necropsies, everyone from the lab pitches in, and one of the key players has been Carly Stark, who also has a poster today, poster number 160. And none of these studies would really be possible without a very dedicated animal care and use committee. So I'll close there and thank you for your time and attention, and be happy to take any questions.
Okay, this paper is now open for discussion.
Irene.
Hi, Jason. I guess I could have asked you back home, but so actually I find very encouraging that antibiotics, at least that particular one, didn't do anything since we use them so much in people. But my question has to do more with the translocation versus no translocation of specific bacteria. And my question to you is, even if they don't translocate, don't you think that the production of metabolites locally may still be affecting things like inflammation, even if they don't translocate?
They absolutely can, yeah, that's for sure. But making a dysbiotic microbiome, at least looking at all of the things, we didn't look at the metabolome, and we're hoping to do that with Adam, to see if VENC does affect the metabolome. But in untreated disease, that doesn't seem to affect how quickly they progress to AIDS. Yeah.
Additional questions? Maybe I can ask one if people are coming. Jason, what about the role of Th17 and the impact? You've done a lot of studies. How do you think the particular bacteria that are there or altered affect some of the immune interactions that you've characterized in the past?
Sure. So there's studies by, his name is Ivan Vučković-Veen, and he's done a lot of analysis of tryptophan metabolism and how that relates to the level of lactobacillus. And there do seem to be relationships between altered tryptophan metabolism and how many lactobacillus species there are. So the question is, how much of that contributes to the depletion of Th17 cells relative to all the inflammation that is occurring as a result of SIV or HIV infection? So it could be additive, but it might not make a significant, in that setting, a significant contribution.
Okay. Any other questions? If not, again, thank you for an excellent talk. I'd like to welcome our next speaker, Dr. Nikki Klatt from the University of Washington, Associate Professor in the Department of Pharmacognosy and Director of the Mucosal Immunology Core. Nikki has made some really important seminal contributions on our understanding of HIV transmission with regard to understanding the role of the microbiome, and she's going to tell us more about those studies today. Nikki, thank you so much.
Thank you, Alan and Elizabeth, and thank you to the organizers of IAS for the invitation to speak. Although my enthusiasm for this is dampened with the huge picture I have to look over to get to the slides, that's just mean. So we're going to go back to the female reproductive tract now in HIV acquisition. So I really appreciate the fact that I got to speak after Jacques because he gave such a beautiful introduction to bacterial vaginosis and vaginal dysbiosis and what this means, and I'll go back to this. But, you know, I first want to just bring up the fact that while we recognize that PrEP is very effective in men and has been used very well in men, in women, this is not the case. So trials of PrEP efficacy in women have really varied in their efficacy from negative efficacy, like in the VOICE trial. Oops, sorry. There we go. In the VOICE trial, where the efficacy was actually negative, to just limited efficacy in the 60 to 70% range with these oral PrEPs, mostly in partner studies with married participants. And then there's also topical PrEP trials like the Caprisa 004, which I'll be talking about mostly today, that had 39% efficacy, and then very limited efficacy in VOICE or FACS. So the question is why are there such varying outcomes from PrEP trials in women? And a lot of this has been attributed to adherence, and adherence is certainly extremely important in efficacy for any drug. But this has been unclear why the adherence would be so much more important in women than men, and if there is actually biological factors that are affecting the outcomes of these trials. So like I said, Jacques gave us a really beautiful introduction of the female reproductive tract and what the microbiome looks like in the female reproductive tract. As he said, and I 100% agree, that bacterial vaginosis or vaginal dysbiosis is probably not the proper term, but it is what is used most highly right now. So I will be talking about BV-associated microbes, but I do think this is something that needs to be redefined as well. So this is the most common cause of vaginitis. It is characterized by loss of lactobacillus species. And as Jacques said, the lactobacillus makes a very healthy microenvironment for the vagina by inducing a very low pH from the lactic acid. And when you see a vaginal dysbiosis or a BV-like state, what happens is that you get an expansion of multiple different, mostly anaerobes and facultative anaerobes. And so predominantly Gardnerella vaginalis, which we'll talk a lot about here, but other bugs like Oediphobium, Prevotella species, and other BV-associated bacteria. It's often associated with vaginal inflammation, discharge, and discomfort, but not always. And clinical diagnosis of BV is not always associated with vaginal dysbiosis, and the same thing where the microbes can be very dysbiotic without having any clinical symptoms. But what is very important, and that has been shown time and again, is that there's an association with increased HIV risk and bacterial vaginosis. And this is true not just where there's an increased risk of HIV transmission to a woman who has BV, but also a man sleeping with a woman who has BV has a higher risk of infection, and it can also increase the risk of maternal to fetal transmission. So there's been a lot of really nice work trying to define why bacterial vaginosis or vaginal dysbiosis might affect HIV transmission. Doug Kwan's group at the Reagan has done really fantastic work looking at how different community-type states can affect inflammation, and of course inflammation is a large predictor of HIV transmission. So we know that there's multiple ways and multiple layers of how dysbiotic microbiome in the vagina can affect transmission. But we really started thinking about these PrEP trials and what's going on with acquisition and prevention, because one of the things that is very clear is that there's increased prevalence of bacterial vaginosis-like states, or this vaginal dysbiosis, in areas that have very high prevalence of HIV infection in women. So this is a nice slide that Adam Bergener made where he summarized different trials from different groups or different studies where they've looked at the different community states. And the different purple colors represent Gardnerella vaginalis, or a BV-like state, where the orange and yellow are lactobacillus. And what you can see in North America, where infection is very low in women, the predominant women have lactobacillus, whereas in Africa, where of course infection is very high in women, the majority of women actually have a Gardnerella vaginalis dominant or a BV-like state. So this brought us back to this question of, does the vaginal microbiome affect PrEP efficacy? And then if so, how does it do that? So Adam uses his very novel proteomic techniques where he does shotgun mass spectrometry, and he's been able to actually identify the vaginal microbiome by using these proteomic-based techniques. So this is actually mass spec based data, not 16S RNA sequencing. But we have shown in a couple different studies that there's a very high correlation between the mass spec-based data and 16S rRNA. But what he did is using the CAPRISA 004 trial, so that we were fortunate enough to collaborate with Slim Cream and the CAPRISA 04 trial, where they used, this is a vaginal microbicide, it was a tenofovir microbicide gel trial, where women were given the microbicide gel to use pre or post coitus. And so from samples from this trial, Adam used mass spectrometry to look at the microbiome, and what you can see is that, yes, there's 4 or 5 community states that, like Jacques and Doug have described very well, but for power-based reasons, we really split these into 2 major states. So either women who have lactobacillus dominant, so over 50% lactobacillus, and you can see, as is typical, if women have lactobacillus, it's almost all lactobacillus, and not polymicrobial, or this polymicrobial state where, there's less than 50% lactobacillus, so we call this not lactobacillus dominant, and this would be our vaginal dysbiosis, or BV-like state. But importantly, the clinical BV was not measured in these women. So just to show the study design from the study, we ended up, after excluding for things like stored sample issues and QC issues, we ended up with 688 participants analyzed from the CAPRISA 004 study. They were evenly distributed between tenofovir gel or placebo gel group, and then we broke them down into this lactobacillus dominant or non-lactobacillus dominant groups. And what you can see is that if you look at efficacy, so remember I showed you that first figure where in the CAPRISA 04, when they looked at all women in the trial, the efficacy was 39%. For every woman. But when you break them up into 2 groups, so women who have lactobacillus dominant over here versus women who are not lactobacillus dominant, something very interesting and very important happens. And that's that suddenly you have a difference. Sorry, this is very sensitive. Suddenly you have a difference in efficacy in women that are lactobacillus dominant. So you actually have increased protection of efficacy of 61%. It gets up to 78% if you control for adherence. And then in the non-lactobacillus dominant group, you don't have any efficacy at all, where you actually have 18% efficacy. So you're losing efficacy if women do not have lactobacillus dominance. So this was very interesting. So and this was demonstrating that, okay, there's differences in the actual PrEP trial based on what the vaginal microbiota was. So we wanted to understand why. And what's interesting is, so like I said, a lot of the data from this was blamed on adherence. So, okay, the women must not be using it. And in fact, when you look at women with non-lactobacillus dominant microbiotas, they do have decreased levels of tenofovir in the vagina. But that doesn't make any sense at all. right? So in a woman who has lactobacillus dominance, you have about 50-50 detectable versus undetectable, but it's 30% detectable to 70% undetectable if a woman does not have lactobacillus dominance. So we ask the question, is tenofovir directly affected by the bacteria? Is something going on here? Because we just don't think that a woman that has non-lactobacillus dominance for some reason is less adherent. So to assess this, we actually went in vitro and did co-cultures using mass spectrometry-based approaches to quantify tenofovir from co-cultures with bacteria. So on the left-hand side here, so in the black is our abiotic controls, and I'm sorry, there must be some timer thing in here because it keeps switching on me. In black you have abiotic controls, where this of course is just the tenofovir and media to make sure there's no media effect or breakdown of the tenofovir itself. And then you have our lactobacillus inners or lactobacillus crispatus in blue, and you can see that the tenofovir is very stable over time in these cultures. But when you have Gardnerella vaginalis in culture, in the presence of the tenofovir, what you see is that you have a significant and rapid loss of tenofovir. So 4 hours, you have a huge drop in tenofovir, and by 24 hours in these cultures, you actually have 70% loss almost of your tenofovir in the culture. What's important, so we're going to focus mostly on lactobacillus and Gardnerella for the purpose of the study because these are the two major bacteria groups. But importantly, we did show that when you look at the other anaerobic bacteria associated with BV, like Prevotella amnia or Bivia, Mobilicus malaris, or E. coli, that all of these are also significantly lost compared to their abiotic control. So multiple bacteria are capable of actually depleting the tenofovir in these cultures. So, but like I said, back to the just Gardnerella and lactobacillus, which I'm going to use for most of the comparisons to the rest of this, what you can see is not only do we lose the tenofovir in the Gardnerella culture, but you also increase Gardnerella inside the bacteria cells, where you do not increase the tenofovir inside the lactobacillus cells. So we asked, is the tenofovir metabolized? So what's happening here? Is it just binding? Is it somehow passively diffusing into the bacteria, and so it's supernatant? Or is this an active metabolic process where the tenofovir is being broken down? So tenofovir is a purine analog, and so we measured putative metabolites, and with adenine being a major metabolite predicted with loss of the oxymethylphosphonic acid. So when we went back to our mass spec approaches to measure these metabolites, what we see is, and this is measuring adenine over here, is that again in Gardnerella you have this rapid and high increase of the adenine production, but not in the lactobacillus. And when we look at the total drug recovery, so this is tenofovir plus adenine, we get the majority of our drug recovered. So this does indicate, one, that the adenine is the major metabolite of this process, and two, that the Gardnerella vaginalis can in fact break down the tenofovir by a metabolic process. So this is all great, but this is all in vitro, and what does this actually mean for in vivo trials? And so what was really important to us is pharmacologically active tenofovir. So tenofovir itself is not pharmacologically active. It has to go into the target cell, and it becomes tenofovir diphosphate, and that is the pharmacologically active version of tenofovir. So we wanted to assess, well, is this actually affecting that process, and is it actually affecting the conversion to diphosphate within the actual target cell? Because if it's not, this doesn't really matter. So we wanted to bring this Jurkat cell component for a target cell here. So now what we have is we have our co-cultures with tenofovir and our different bacteria, Gardnerella, lactobacillus, and then we also add Jurkat cells, which would be the putative targets for HIV in our in vitro system here. And then we go through a series of measurements, so looking at loss of tenofovir in supernatant, indicating metabolism or target cell uptake, looking at the presence of intracellular tenofovir diphosphate, so this indicates the target cell can take it up, and then we want to look at that versus how much adenine is made, because if the adenine is made faster than the tenofovir diphosphate, then we know that the bacteria are kind of winning this race with the target cells. So to look at these kinetics then, we have the tenofovir alone in black, so you'd expect that to stay high. And now we have, and so when it says cells here, this means our Jurkat cells, right? So in blue again, we have our lactobacillus inners or crispatus in the presence of Jurkat cells, and you can see the tenofovir goes down, which you'd expect because, you know, hopefully those target cells are taking up the tenofovir and turning it into tenofovir diphosphate. However, when you look at the Gardnerella in the presence of the Jurkat cells in the tenofovir, you have an even more steep decline in the levels of tenofovir in the supernatant. And then again, as we showed earlier, you get this adenine formation in the presence of Gardnerella, which you do not see with lactobacillus or abiotic controls. So probably the most important part of this, though, is, like I said, is that tenofovir diphosphate being actually produced in the cells that would actually protect those Jurkat cells from infection from HIV. So when we look at either tenofovir alone with the cells or the tenofovir plus the lactobacillus, you see this rapid rise in tenofovir diphosphates. That's very important. So this would be pharmacologically active and presumably would protect the cell from being infected. However, when we look at the Gardnerella vaginalis cultures, you see a significant decrease in the diphosphate production. And again, we look at this total drug recovery, which importantly shows that we are getting all these metabolites back with the adenine versus the diphosphate versus the tenofovir. So finally, this is kind of the same data, but just to show it in a little more clear picture and really highlight how rapidly this happens. So here we have our tenofovir in the presence of Jurkat cells, the red again in the vaginalis, and the blues are our lactobacilli. So just in 15 minutes, you already have the significant decrease of the tenofovir. So this is tenofovir diphosphate to tenofovir ratio. So pharmacologically active tenofovir relative to not active tenofovir. And you can see this is decreased as quickly as 15 minutes. 30 minutes, it's even is more significant. And by 1 hour, you really have this substantial decrease in the tenofovir diphosphate compared to lactobacillus or the tenofovir alone. So these really data highlight that not only are the Gardnerella metabolizing the tenofovir, but they're actually metabolizing it faster than the target cell can uptake it and make it pharmacologically active. And we've done a lot of work on the kinetics of this and what things affect it, like lactate, which also was highlighted in Yak's presentation. But my grad student Ryan Chu has a poster tomorrow, and where he is going through a lot of the other kinetics into even further detail if you're interested. So to conclude, the vaginal microbiota impacts microbicide efficacy of HIV acquisition in this tenofovir gel trial. The BV-associated bacteria, as Gardnerella and Prevotella, rapidly metabolize tenofovir to adenine, and the active tenofovir diphosphate is decreased in the presence of the BV-associated bacteria, which was more rapid than the target cells can uptake it for protection, which indicates that this is a potential mechanism for microbiome-induced reduction of PrEP efficacy. It also highlights the importance of both adherence as well as vaginal health for PrEP efficacy against HIV in women. And I do want to highlight that we know it's not just metabolism. These vaginal dysbiosis can cause all these other inflammatory sequelae that can also affect HIV transmission. I just want to highlight some future studies we're working on and the importance of this in other aspects of HIV infection. So there's many other prevention drugs in the pipeline, and there's candidates for going in for drugs. And so we're doing a lot of work trying to understand the role of bacterial metabolism with other drugs, and we do see some very interesting metabolism in some of these drugs that hopefully we'll have those data out very soon. And then also the GI tract microbiome, there's a lot of bugs in the GI tract. You know, Jason just talked about how important these are, and what we know is that a lot of the GI tract bugs can also metabolize antiretroviral drugs. And so what does this mean for cure or viral reservoirs in HIV treatment? So finally, I want to thank my lab, particularly Ryan Chu, my really phenomenal graduate student who spearheaded all these mass spec assays to look at the drug kinetics, as well as Alex Zevin, a very talented postdoc who is my microbial expert. And then, of course, Adam Bergener and his group, phenomenal collaborators, and they spearheaded all the mass spectrometry approaches, CAPRISA 004, and of course CAPRISA and SLIM and all of their support and samples. And I'd be happy to take any questions.
This paper is now open for discussion. Any comments or questions for Dr. Klatt? Maybe while people are coming to the microphone. Nikki, what about other inflammatory mediators and how those affect in there? What's the effect or dual effect of not only the bacteria, but then the inflammatory effects as well that are coming up during these?
So this is a really good question and a really important question, and we're just not sure. So we haven't teased that out exactly. So Adam has, you know, one of the really powerful parts of doing these proteomic-based approaches is that he doesn't just get the microbial proteins, but he also gets the host proteins. So he, I know, has a lot of interesting data coming out of his lab about inflammation and the epithelial barrier, because we also have seen quite a bit of epithelial barrier regulation from the bacteria, and showed in a recent paper last year in PLOS Pathogens that Gardnerella can actually decrease wound healing abilities in the epithelium. So I think that all of these mechanisms are probably at play, but trying to tease out exactly how much each one has been very difficult. And so future work will have to be done on that. Oh, good, they made the picture bigger.
Yes.
I apologize. Gustavo.
Gustavo.
Thanks. Great Great work. Did you have to do any antiviral activity? Did you challenge the cells?
So great question, and Ryan I know is working on that right now, but I'm not sure that I've actually seen the data because I know he's got lots of cultures underway. And so we're really focused now. We're moving into dapivirine and TAF and some of the other drugs, and so I know he's added that component, which is a very important component. So.
Well, thanks again, and I think we'll move on to the next speaker. Our next speaker is Dr. Rupert Kaul from the University of Toronto. Rupert is the director of the Clinical Infectious Disease Group at the University of Toronto, has extensive work also in Nairobi and Kenya, and we'll be talking today about the therapeutic approaches for the microbiome and opportunities we might have there. Rupert?
Thanks very much. I'm still seeing Nikki's much better data than mine here. Push which? Oh, press escape. Control, escape. Okay, gotcha. Then just click on mine. Yeah, I'm getting there.
Talk again, that's fine. Just click on it.
Oop.
There we go.
No, it's pulling up CLAT again. No, escape, for God's sake.
Come on, I'll put it up for you. There, okay.
Okay. Thanks very much. So thank you very much to Vic Appé and the organizers for inviting me to speak. I would like to thank the session chairs for allowing me to present my data, but they told me I wasn't allowed to, and that I had to review clinical trials to improve the microbiome function in HIV. So, which is a lucky thing anyway, because the data of my co-speakers has been much better than any data that I could give you. And I do sneak a bit of my data in anyway, of course. So I think when you, as a clinician, I think it's important to take a step back and say we've seen a lot of very interesting descriptive studies about differences in the microbiome. As a clinician, obviously, I'm interested in saying, okay, for somebody that I'm working with or providing care for in terms of their HIV infection, what can I do to intervene here, and what difference is it going to make to that person? And so I appreciate there are two different ways you can look at the function of the microbiome. You could say the microbiome functions by making butyrate and short-chain fatty acids and things like that. I'm really talking about function in terms of the function for the organism, that is the person living with HIV who's sitting in the chair in front of me. So we think that the function of the microbiome is multiple, and I'm only going to be focusing on a couple of things in the talk. So clearly there are influences that have been shown on neurocognition that are very interesting, on mood disorders, on obesity, on reproductive health, pregnancy outcomes that I'm really not going to be focusing on. I'm really going to be focusing on, I think, the most immediate and obvious impact that the microbiome has is on local immunology, so the immunology right there at the site where that microbiome is active. Influences more widely on systemic immunology. And I think that clearly we've heard in the review that there is a lot of assistance of the microbiome in mucosal defense, so that lactic acid is repelling genital pathogens, cervical mucus, as Jacques was talking about, can trap HIV. There's a super neat paper that I encourage people to go and have a look at, talking about how specific microbial colonizers on the skin can produce antimicrobial peptides that can then directly repel pathogens, so pathogenic Staph aureus, for instance. And again, I'm not really going to be talking about the skin in this talk either. So the next question is, and again, luckily people have already introduced you to this, how has the microbiome changed in the HIV context? And I'm going to be focusing on two things. So one is people who are living with HIV. There's been a focus, and Alan talked about it, how the gut microbiome changes. In particular, we have a decrease in butyrate production, reduced diversity, increase in proteobacteria, decrease in the firmicutes. Microbiome's also changed in a number of other areas that I think are probably less functionally important to my patients, so I'm not going to be talking about those as much. In general, those differences in the gut microbiome are improved when we put somebody on effective antiretroviral treatment. It may be there's some data that the integrase inhibitors may be better, but certainly those things improve over time. People who are at increased risk for HIV then, or HIV acquisition, there are clearly differences in the genital microbiome. There may be differences in the rectal microbiome, as Jason touched on. We don't really know that yet in terms of having HIV acquisition as an endpoint. But certainly the penile and the vaginal microbiome has been shown to be, differences have been shown to be associated with HIV acquisition. In particular, in both vaginal and penile microbiome, increase in gram-negative anaerobes, particularly Prevotella, and in the vaginal microbiome, decrease in L. crispatus. Again, some nice work from Doug Kwan that Nikki alluded to is associated with an increased acquisition of HIV. But there are all kinds of possible confounders, and again, I think that, again, Jason introduced us to that very well. So receptive anal intercourse has been shown by 2 different groups now to have major impacts on the rectal microbiome. Of course, somebody who's more sexually active and therefore at more risk for HIV is more likely to have co-infections that can also change the microbiome and themselves are part of the virome, potentially, if we're talking about herpes or HPV. Antiretrovirals have been shown to have direct antibacterial effects, so can change the microbiome of the gut. Antibiotics, diet, smoking. Rebecca Brotman has some nice studies looking at how smoking can change the microbiome, alcohol, etc. So there are many potential confounders that may lead to these descriptive changes of microbiome alterations in association either with HIV acquisition, HIV risk, or HIV infection. So if we're wanting to do clinical studies, then what would be the outcomes that we should look at in those trials? So I think that if you're talking about people living with HIV, really we're talking about systemic inflammation, this inflammaging that was touched on by Alan, that can lead to increased coronary artery disease, lipohypertrophy, the non-alcoholic steatohepatosis or non-alcoholic fatty liver disease, and the HIV-associated neurocognitive disease. But again, in general, the studies that we're going to be looking at first probably aren't going to have those less common clinical outcomes. We're probably going to be looking at actual systemic inflammation, and that is what most of the studies out there have done. If we're talking about people at risk, we know that mucosal inflammation, I don't have time to really go through the data, but there's very nice information that both in the male genital tract and the female genital tract that inflammation is associated with increased HIV risk. So again, I think microbiome alterations, the functional impact of those presumably would be mucosal inflammation, which would be increasing your HIV risk, and potentially direct acquisition of HIV or of other sexually transmitted infections. So, and again, I think it was nice, I'm not actually talking about postbiotics because I don't think there are any clinical trials yet that have looked at postbiotics functional outcomes. But we could selectively remove bacteria in our clinical studies with an antibiotic. We could selectively replace bacteria with a fecal microbial transplant, FMT, putatively with a vaginal microbial transplant. And there are actually studies saying that again, same-sex couples can share vaginal microbiomes, some nice work that came out a number of years ago. And I don't know if other people work with obstetricians. An obstetrician in Toronto, certainly when she had her own baby, by C-section wanted to paint that baby with vaginal secretions because the vaginal microbiome was felt to be important for neonatal health. And so again, could a vaginal microbial transplant be something in the future? There's certainly no data yet. And probiotics. And you can support growth of certain bacteria either with prebiotics or by altering the environment in different ways. So as Tom Hope would probably say, okay, enough pissing and moaning, just give me some data. So I'll go on and give a brief overview, and I apologize in advance to anybody whose studies I'm not presenting. I'm sure there are many studies out there that I should be discussing. Just come up and give me a kick afterwards. So in terms of pathogenesis, when I talk about pathogenesis, I'm generally talking about people who are living with HIV, what studies have been done to try and alter the microbiome, and what have the impacts been of that in terms of often systemic inflammation as the readout. So there certainly was a nice clinical study done by Vesterbach and her group that looked at initiation of Septra, showing that if you started Septra, so a sulfonamide-based antibiotic that's often used in Africa in conjunction with antiretroviral therapy, that reduced markers of bacterial translocation, but as far as I know, they didn't look at inflammatory biomarkers in that study or other clinical endpoints. And so I don't think there's that much looking at antibiotics in humans and the impacts on systemic inflammation that we're interested in. There have been a couple of probiotic randomized controlled trials. So the two that have come out and have been published, one looked at a combination of lactobacilli and bifidobacteria. This was the Stixford group in ART-treated individuals. The ends that I put in here are always the number who got the active drug versus the number that got placebo, just so you can get some idea as to how big these studies were. So a relatively small study. In that study, they saw a decrease in D-dimer a procoagulant effect, a factor that goes along with inflammation, a decrease in C-reactive protein, and some microbiome changes as you would expect. A study done looking at S. boulardii showed a similar effect in terms of some microbiome changes, a decrease in LBP, the LPS binding protein, and IL-6. In again, quite small studies with some dropout in those studies, so perhaps not ideal follow-up. There are 2 studies ongoing looking at Vaginal. One is an ACTG study that will be done much quicker, and Alan is involved in that. The second is a Canadian-funded study that I'm running in immune non-responders, and the ACTG study is looking at a wider swath of people who are taking antiretroviral treatment. So stay tuned for the results from those. Those are ongoing, and I'm not going to bore you by talking about the setup of those studies. In terms of prebiotics, there are a couple of neat studies looking at prebiotics. So one looks at, and I'm not going to try and go through the what type of oligosaccharides they're using, and I don't know enough about the prebiotics, but essentially giving prebiotic is giving food that should support the growth of certain bacteria and give them a selective advantage over putative pathogenic bacteria. So 2 studies have been done that I'm aware of looking at that. One gave a combination of these oligosaccharides. They enrolled participants both not on treatment with a good clinical response, and on treatment but with a slow clinical response. And they saw, as you would want to see, an impact on the microbiome, an increase in diversity, a decrease in the proteobacteria, and an increase in the Bacteroides. And also saw a reduction in some of the antimicrobial peptides systemically, reduced T-cell activation, and increased in C-reactive protein. When you look at another oligosaccharide mix, you see fairly similar changes in the microbiome, a decrease in soluble CD14 and an increase in NKT cell function. And I think what you're starting to get is, again, I'm primarily an immunologist in terms of the research that I do. There are many immune parameters that I can look at, and what we're seeing, I think, is that people, when they do these clinical trials, tend to look at a lot of immunological factors and pick a couple that change when you do your study that may change in favorable ways. And I think that's something we probably need to do as a field, is to be more clear about what the key immunologic endpoints are going to be and standardize our readouts a little bit to enable, I think, someone like me who comes to review these studies, and you who have to listen to it, apologies, enables you to get a clear idea of what's going on. In terms of fecal microbial transplantation, there's just one from Mar Somsok's group very recently down at UCSF that is, I think it's still the in-press publication that's online, looking at fecal transplant. And again, I don't know as much about fecal transplantation, the open biome system. So open biome is a way that donors can provide a healthy stool, putatively healthy stool for fecal transplants. And these potential donors were screened to have low proteobacteria, high Bacteroides, so a stool that you would think might reverse some of the changes you would want to reverse in the context of people living with HIV. They gave a single administration via colonoscopy. Mar's a GI specialist by training. There were 6 in the active group, two who received placebo, and there was a moderate impact on the microbiome without any immune impact on the parameters that they'd quite rigorously defined in advance. I think it's a summary, and I think they're thinking about where to go with those data. I don't want to speak for them. So moving on, and many of you probably are wishing that you were drinking wine in a French bistro. You'll have to go to California to get this Cabernet Sauvignon, the BV Cabernet Sauvignon. But moving on to the vaginal microbiome, there have been, again, it's hard to know what function we should be looking at. There have been a number of studies, of course, looking at BV treatment. BV treatment's a frustrating process, so as a clinician, if we treat somebody's bacterial vaginosis, much of the bacterial vaginosis out there is asymptomatic. If we do treat it, it tends to reoccur. The treatment isn't that effective, and it tends to reoccur relatively quickly. So there have been 3 clinical trials with an STD acquisition endpoint, which I think is an important functional outcome if we think that part of the role of the genital microbiome is to protect against STI acquisition. In one where asymptomatic BV was treated for 5 days and then suppressed twice a week with topical flagyl, metronidazole, one of the classical antibiotics we use to treat, a reasonably sized study. And in that study that Jane Trebke did back in 2007, there was reduced STI acquisition, particularly chlamydia, at a 6-month endpoint. However, her follow-up study, which came out just recently in ClinID, where women who had a bacterial vaginosis were treated at baseline, did home screening every 2 months, and if they were found to have bacterial vaginosis, were then treated up front with Flagyl, was found to have no impact at all on STI rates. And this was a very large, well-powered study. So I'm not sure where to take that. There was a modest reduction in bacterial vaginosis, about a third reduction in bacterial vaginosis. And Scott McClelland and his group have shown that in their trial of topical Flagyl and meconazole to try and prevent both candidiasis and bacterial vaginosis, there was again a reduction in STI in high-risk women in Mombasa who were not selected on the basis of bacterial vaginosis specifically, but may also have had candidiasis. What about antibiotics? So the only thing that I'm aware of looking at a functional outcome in terms of antibiotics is a study that Vineet Jog in my lab has just wrapped up, and we're in the process of writing up, and thanks to Jacques in advance for helping us with the microbiome. This is just an observational pilot study looking at Flagyl therapy in HIV-negative Kenyan women, 45 Kenyan women with BV, most of it asymptomatic. And here the primary endpoint that we selected was actual pseudovirus entry into cervical cells when we took a cervical cytobrush 1 month after treatment. And some of the secondary endpoints that I'll share with you are genital cytokines. So first of all, when we treated bacterial vaginosis, as in many studies, only about half the women were BV-free after a month. Overall, there was a significant but modest, I would say, reduction in virus entry into CD4 T cells from the cervix, which was our primary endpoint. But there are some neat cytokine results I just want to share with you. So if you, in both of these upcoming figures on the left, I have the overall. This is the entire group at baseline and after a month after treatment. This is the roughly half the women who cleared their BV, and this is the roughly half the women who did not clear their BV by Nugent score when we do a Gram stain on a slide. So what you see is that in the women who clear their bacterial vaginosis, there's quite a dramatic reduction in IL-1 alpha and IL-1 beta, two of the prototypical pro-inflammatory cytokines. In the women who don't clear bacterial vaginosis, as you might expect, there's no change at all in those cytokines, and there's actually no change in pH or any of the other parameters that we looked at. Interestingly, if you look at a number of other cytokines and chemokines that have been associated with HIV acquisition risk in the Caprisa data, so Nikki presented some great data from the Caprisa trial. That same group, Joanne Passmore and her colleagues, had a paper a couple of years ago showing that certain overall inflammation and certain inflammatory cytokines and chemokines were associated with increased risk of HIV acquisition. IP10 was the one that was most strongly associated. So after successful treatment of bacterial vaginosis at 1 month, there's a dramatic increase in IP10 levels. No change again in the group where bacterial vaginosis was not successfully cleared. And we see that for a range of inflammatory cytokines and chemokines, including, as I say, MIG, MCP, IL-17, MIP3 alpha, a number of chemokines that have been associated actually with a pathogenesis of HIV transmission at a mucosal level. That's kind of interesting. Now, we have not followed this up. We would be interested to do a larger study that follows this up in more detail, so we don't have more time points to know how long might this be sustained. Is this a transient thing or is it going to go away? But I think it gives us a little pause in thinking about whether we should be rolling this out at a population level. And again, I think in terms of probiotics overall as a cure or as an adjuvant therapy for bacterial vaginosis, this is just a nice meta-analysis that I would send people to, suggesting that probiotics may have some benefit, but there's a lot of heterogeneity in the studies, so it's hard to know to what degree a probiotic can help. Our existing probiotics can help. I lead into that because I think it's important to, when we're treating bacterial vaginosis, not just to treat that bacterial vaginosis, but potentially to restore the right bacteria in that person's vagina as she moves forward in terms of reducing her risk in the future. This is just reiterating the data from Doug Kwan's study, the FRESH cohort in South Africa, a cohort of women at high risk for HIV acquisition. And I'm colorblind, so I hope I say the colors right. But this one on the bottom, where they saw no HIV acquisition, were women who had a Lactobacillus crispatus predominant microbiome, which is not the norm in African and Caribbean women, so either South African black women or African and Caribbean women in Toronto, where we've done similar studies. So it's quite rare to find a crispatus predominant microbiome, but none of those women acquired HIV. The Gardnerella, the sort of bad, what we would think of as being the bad for HIV risk microbiome, the more dysbiotic microbiome was associated with HIV acquisition, but there was a near significant association of Lactobacillus iners, that is the predominant Lactobacillus species in black women both in South Africa and in Toronto, where we've done studies with HIV acquisition. So presumably restoring a Lactobacillus iners flora might not be a healthy thing to do. Most of the probiotics that we use actually at the moment don't include vaginal bacteria. They include lactobacilli that are often found in the gut or other places. They're not actually naturally present in the vagina. So one question is, how can we restore Lactobacillus crispatus predominant microbiome? And I just want to point you as a future direction. Craig Cohen and his group from UCSF are doing a neat clinical trial at the moment of Lactin V, which is a Lactobacillus crispatus-based intervention probiotic to reduce BV recurrence. So again, it's a phase 2b trial that's ongoing at the moment. The primary endpoints are safety and BV recurrence, and I think that might be a useful tool. We'll see when the results come out in September of 2017. So finally, I thank Mario Ostrowski for this slide. This is apparently a true prank that was played on the church just down the street from him in Toronto, where coming up for Christmas time, somebody changed around the letters outside the church to join us tonight for Messy Foreskins, Amen. So Mario promised that he did not do that, but thank you, Mario, for sending it to me. So do we have any interventions in terms of the penile microbiome? So not really. I think I just want to highlight, we know that circumcision reduces HIV acquisition in 3 clinical trials, and Cindy Liu and her collaborators, of whom I'm one, so I'm biased in terms of presenting these data, have shown that circumcision, as you might expect, because it opens up the previously anaerobic space under the foreskin to the air, is associated with a dramatic shift in the microbiome away from anaerobic bacteria, including Prevotella, and towards standard skin anaerobes. And that those same bacteria that are lost after circumcision, if you take men who remain uncircumcised and follow them prospectively and say, okay, what microbiome is associated with HIV acquisition, a foreskin microbiome in the subpreputial space that is enriched for really the same bacteria we see associated with risk in women, so Prevotella and other gram-negative and gram-positive anaerobes. Many of those anaerobes are themselves associated with increased HIV acquisition risk, and actually associated also with inflammation and local levels of IL-8 and other inflammatory cytokines. So that paper, I think, just came out today, actually, in MBio. There were some neat studies that have looked at, so first of all, there have been a number of clinical studies saying if I'm a male partner of a woman with bacterial vaginosis, does it help giving me antibiotics? And the short answer is no, it does not improve her chance of having reduced recurrence of bacterial vaginosis. I just wanted to highlight one study that's quite interesting from Elizabeth Bakusi, where in Kenya, the male partner was randomized to a topical microbicide. It was actually Purell, like an alcohol gel, that he cleaned his penis with every morning and then before and after sex. And actually in that, and the woman had her bacterial vaginosis treated, and in that study there was actually increased BV recurrence, or a trend to increase BV recurrence in the partners of men who used the Purell for penile hygiene in that context. So certainly I think there's a lot of work that needs to be done in terms of how to reduce a woman's risk of bacterial vaginosis, or to shift her microbiome, if you will, from a microbiome characterized by diversity to a microbiome characterized by L. crispatus, which is what we would like to do. So just to summarize, there are some strong associations between the microbiome and HIV susceptibility and pathogenesis. I think that induction of mucosal and systemic inflammation is thought to be central in terms of the mucosal for the HIV acquisition risk, and the systemic for the and negative outcomes in people living with HIV. There are a lot of potential confounders. So far, the trials have been pretty small, and I think because of those confounders, it's really important to get some human clinical trial data where we can really tease out what is a true effect of the microbiome and what is potentially relatable to some of these either known or unknown confounders that I think are important when we're studying the microbiome. So I just want to thank Vineet, who did that trial of bacterial vaginosis and immunology, and Rodney, who is running our clinical trial of probiotics. And thank you for your attention.
We're running a little late. I'll leave it to the end here. Thank Rupert for an excellent question. Any questions for any of the speakers, please come up front. We'll be here for a few more minutes. And thank all the speakers for really an excellent session. Big round of applause for everybody.